Welcome back to Study Room with your host, Catwoman and Cat. Today's topic is transcription. So before we talk about transcription and the different elements that are needed in transcription, first let's know the definition of transcription. Transcription is the action or process of transcribing something. In genetics, we're going from a double-stranded DNA to a single-stranded mRNA strand, where mRNA leaves the cell nucleus and enters the cytoplasm. So a chromosome is a long molecule that has millions of base pairs in humans. To be exact, there's 3 billion base pairs. But, however, certain parts of the chromosomes is important for our bodies, known as genes. Gene codes for different things, and a gene that is being expressed will produce a specific protein. How? This occurs by transcription, which is the process of an enzyme uses one DNA strand to produce a template known as mRNA. And it occurs in 3 steps: initiation, elongation, and termination. The initiation happens at where RNA polymerase binds with the help of initiation factors at a specific sequence with a gene known as the promoter sequence, which splices the 2 strands apart. One of the strands will be the template strand, also known as a antisense strand. This strand will become the mRNA. The other strand will become the non-template strand and become the nonsense strand. So RNA polymerase will go down the DNA and continue adding the mRNA sequence until it reaches a termination sequence, and that's when RNA polymerase releases. So we have to talk about the difference between DNA and RNA, right? In RNA polymerase, it doesn't require a primer. The RNA reads the sequence from 3 to 5, but it's built from 5 to 3. Also, RNA is ribonucleic acid, which uses ribose sugar rather than a deoxyribose sugar. And it expresses uracil other than thymine. This video is just to help give you a small idea of what occurs in transcription. Once you get a basic concept, you can move on into a more complicated method. But first, focus on the small steps, and from there, you keep adding more of the initiation factors that are needed and the specificity of the RNA polymerase, what is the promoter sequence. So for right now, just know that those 3 steps are initiation, elongation, and termination. We have RNA polymerase, which binds to the DNA strand and at the promoter site. Thank you so much for being with me today at Study Room with your host, Catwoman and Cat.